Hello, everybody, and welcome to our mini lecture on the ABO blood group. By now, in Module 4, you have been exposed to various forms of inheritance. As we covered in the Darwinian lecture, the finches and all of the different features of the finches really matter in terms of their adaptation to various environments. The interesting complexities of this also lie underneath one's skin or feathers if you're studying birds. With humans and some other animals, we have what's known as the ABO blood system. The ABO blood groups are one of 30 different blood groups that we have in our bodies. This is the most commonly known and the most commonly talked about at the 100 level as the great example of polymorphisms, where two or more alleles exist within a population of individuals. So we are going to talk about inheritance factors of the ABO blood groups. So I encourage you to make sure you go to the lecture in this module and review this on your own. So let's start with the genotype, which you know is the genetic makeup of the individual. The genotype for the ABO blood group will be as follows. AA is homozygous dominant. AO is heterozygous. BB is homozygous dominant. BO is heterozygous. So as you see here, there are the four: AA, AO, BO, BB, homozygous recessive for O, and something referred to as codominant for AB. As you've learned, genotypes code for phenotypes of given features. So if we were going to talk about this in the sense of how it codes, you should know that AA or AO would be the genotype for phenotype blood group A. BO or BB would be the genotype that would code for the B phenotype. OO, as you probably assume, would code for the phenotype blood group O. And this is the only recessive blood group within the ABO. And then finally, we have something called AB, and that genotype is codominance, because the A allele in this case and the B allele in this case are dominant alleles. That code would mean AB is codominant. So as you infer, the four blood groups are A, B, AB, and O. When you're trying to figure out inheritance, you would want to know what the individual's genotype would be to determine the possible outcomes of the individual offspring. So if mom had AO as her genotype and dad had BO as his genotype, those would be heterozygous. So those are two heterozygous genotypes, as you've learned. So what we do to determine the possible outcome of their offspring is create a Punnett square. So these Punnett squares, this could be dad, this could be mom, and then what would go in the first box here? If you said AB, you would be correct. And then in this group, what do you think it would say? BO would be correct. In this box, you're going down, and then horizontal would be OO. And then following that same pattern, AO. So the statistical probability, if I was to ask you this in a quiz, what is the statistical probability of one of their offspring having O blood? Well, you would know the only genotype, as we covered for O, is homozygous recessive OO. So the correct response would be, for statistical percentage, 25%. These Punnett squares are really helpful in determining possible outcomes, but something to remember, as with ABO or many other monogenic or simplified genotypes, you must know the genotype prior to doing the Punnett square. So when you're determining possible outcomes, knowing that genotype will help you determine the possibility of the phenotype. We will continue with this in the next lecture regarding lactose intolerance. Looking forward to it.